hey, what's the dependent variable? So like experimental questions in data analysis are always fair game regardless of what unit we're in. That means I've got 10 minutes left.
Are we gonna have any questions like that bonus, like FRQ about breast cancer and like how we're talking about like observing data like that and
Not at that level. That would be too intense for an FRQ. Like if I give you a graph, you won't have to struggle to figure out what the graph, like what we're actually measuring. Like if it's a graph, it'll be a graph of mitotic index, or it might be a graph. It might be a graph showing you like how much activity a certain kinase has in the cell signal pathway. But like, you should know, if I show you a graph of how much kinase activity there is, you should immediately think like, oh, this is a question about basically how strong is the signal transduction pathway. If the kinase activity is high, the cell is being stimulated strongly. If the kinase activity is low, then it's not being stimulated strongly. Yeah. What else?
We have that one graph that was on the bonus.
With breast cancer? Like
Yeah,
the breast, oh, Western blot?
Yeah.
No. Because it was optional. I wouldn't do a Western blot. You'll start seeing Western blots in Unit 6. Like, you'll be evaluated on Western blots in Unit 6. Yes.
Can you talk about cyclin and CDK?
Yes. So, how does the cell even get to this point where it's in M phase? And a big answer to that question is the interaction between cyclin and CDK. So the example that I gave you, and you don't have to have this specific example memorized, but there is a CDK that is present in the cell that if it's active, it's gonna push the cell through G1 towards S phase. But currently it's not active because it's missing a critical component. It's missing this piece right here called cyclin. Where is the cyclin? Well, it doesn't exist yet, but if the cell is stimulated to grow, this could be an RTK, so I guess I should draw it like this. If the cell is stimulated to grow because a growth signal activates RTKs, and these RTKs phosphorylate each other, and that activates the Ros-Roth-Mek-ERK pathway, which you don't have to, that's the one that you don't have to know. But as a result of this pathway that's activated, one of the genes that is transcribed is the gene for cyclin. This is a type of cyclin called a G1 cyclin because it helps the cell progress through G1. That cyclin can bind to and activate CDK. Now in the example that I gave, one of the things that this CDK does is there's a protein called RB. And RB is attached to and inhibiting a transcription factor called E2F. So RB is inhibiting E2F. CDK is a kinase, so like all kinases, it's going to phosphorylate something. CDK is going to phosphorylate RB and deactivate it. You need to be able to interpret an inhibit sign. So like if B inhibits C, but A inhibits B, A activates C.
What are you talking about?
Right, this is where it gets confusing. That's the same thing as saying, That's the same thing as saying A activates C. So CDK helps activate E2F because it inhibits the inhibitor of E2F. If you see two inhibit signs together, right next to each other, you can collapse it into an arrow. A ultimately activates C. CDK ultimately helps activate E2F because it inhibits RB. How does it inhibit RB? It inhibits it by phosphorylating it. I could measure the amount of phosphorylated RB that exists in a cancer cell to try to get an idea as to whether or not this pathway is working properly. What would you expect in a cancer cell? Because when RB is phosphorylated, that E2F is able to enter the nucleus, and that E2F promotes the expression of even more proto-oncoproteins. And so this basically triggers a surge of proto-oncoproteins to help push the cell through G1 and get ready for S phase. So if I suspect that a cell is cancerous because of some alteration with RB, would I expect that cancer cell to have relatively high levels of phosphorylated RB or low levels of phosphorylated RB?
High levels.
I would expect high levels of phosphorylated RB. Or maybe the RB is mutated and can't even bind to E2F. It doesn't have to be high levels of, but if it comes down to your options being would I expect the phosphorylated levels of RB to be higher or lower? Well, based off of this figure, I would expect a cancer cell to have higher levels of phosphorylated RB, which means stronger production of proto-oncoproteins. And then the other thing that you need to know about cyclin is that the G1 cyclin will eventually be destroyed because it will be marked with a small protein called ubiquitin. Multiple ubiquitin proteins will be stuck onto the cyclin, and that will flag it for destruction. And so what that means is that cyclin levels rise and fall throughout the different stages of the cell cycle. So you have like a G1 cyclin, and as the cell moves into S phase, you have an S cyclin that is produced, and then as the cell finishes S phase, you get a G2 cyclin that's produced. And as the cell finishes G2, you get an M cyclin that gets produced. And as M ends, that M cyclin is degraded. And so that's where these proteins get their name. They cycle up and down in concentration. And it's important for the cell to coordinate which cyclin gets made to make sure that it progresses through the cell cycle in the proper sequence. If a cell accidentally just starts making M cyclin out of nowhere, it might try to do mitosis even though it's not ready to do it.
So each part is for one checkpoint, like you'll go down and up for during a checkpoint?
Yeah, you can think of the cyclins as like helping the cell get past the friction of the checkpoint and on to the next phase. Yeah. Basically, you can think of these cyclins helping to deactivate the tumor suppressor proteins that are trying to prevent the cell from moving past the
Doesn't it need
checkpoint.
to be a growth signal?
Yes, there needs to be a
growth.
there's supposed to be a growth signal to trigger this whole sequence of events. But what if it's a cancer cell and the receptors click together even when there's no growth signal? Well, that would be a problem, right?
That's for every checkpoint there has to be a new growth signal? Or
No, sorry, no. There needs to be a growth signal to start this party. But then once the party is started, most of the signals are coming from the cell internally by the production of cyclin. And then, like, just to finish it off, remember, once the party gets started, that doesn't mean it's guaranteed to happen. If at any point, like for example, DNA damage is detected, or the cell doesn't have adequate resources, tumor suppressor proteins can be activated that inhibit either the cyclin or inhibit the kinase And in that case, it's kind of like slamming on the brakes. And something's gotta happen, like the cell can't brake forever. Once the brakes slam, the cell either has to correct whatever conditions cause the brake, or exit the cell cycle altogether, like give up and say, nope, forget this, I'm just gonna go back to G0. Or it will die. There are some brakes that are, like, if a cell, like G2 brakes, basically after the cell has copied its DNA, there's no going back. You either divide or you die. Like, a cell can't just sit there with twice as much genetic material and go back to G0. It's like do or die at that point. What else? For the small amount of time we have left.
Could you, if you pull up like the AP classroom, there's like one question.
Oh, what's your AP classroom question?
It's number 10 on the unit or topic question.
On the progress check or the topic question?
Topic.
Okay, I just wanted to make sure you. If I can find it quick enough, yeah. Topic questions. Okay, I got it pulled up. Number 4. I don't have time to project this, but. Okay, so this is a question that looks at the epinephrine pathway, which was like one of the first pathways that we looked at, where the hare was running from the wolf. And basically it's saying if a cell is stimulated by epinephrine, normally that epinephrine causes a lot of CAMP to be produced. And that CAMP activates PKA, and PKA activates B kinase, and B activates C, and C activates D, and ultimately the muscle cell responds to that by performing glycolysis. Because a muscle cell needs to do glycolysis to help the hare run away from the wolf. Well, the question says, normally this CAMP gets broken down. What if you inhibit, it says in the question, PDE is an enzyme that takes cyclic AMP and breaks it down into AMP, and that shuts this signal down. But what if you administer a drug X, and it inhibits PDE? What's gonna happen to CAMP levels? This is for anyone to answer. CAMP levels will stay elevated, which means basically the muscle cell is glitched out in this epinephrine signal state. It's not going to be able to shut the signal off. So the correct answer to the question is the enzymes involved in the transduction pathway will stay in an activated state because the signal is basically locked on.
I thought that.
I
know that the conversion of AM stops CAMP.
The conversion of,
Like with PDE.
yeah, when PDE destroys CAMP into AMP, that normally shuts the signal off.
I didn't get that.
But because CAMP levels stay elevated, all of these kinases will stay activated. Which is what they tried to do with Lila when she was in the NICU with her lungs. They gave her a PDE inhibitor called Norinone. Okay, that's all I have time for.